My speaker is not working properly. Can somebody confirm you can hear me? Can somebody confirm they can hear me? If somebody
Yep,
doesn't,
we can hear you.
all right, great. I was gonna kill it and restart again. All right. It said when I came on that my speaker wasn't working. Before we begin, are there any questions? Let's close that. All right, today is January 18th, and we're supposed to be discussing Lab 2, aseptic transfers. I should mention, I forgot to mention this to the other class, that you should read the textbook chapter through the section on compound light microscopy. And you should read that before beginning Lab 3 and Lab 4. Let's go to lab. Whoop, not quick enough. There we go. Why is that so big? All right, any questions before we begin? If not, I'll begin Lab 2, aseptic transfers. Upon completion of this lab, you should be able to, one, understand the purpose of using aseptic transfer techniques, two, be able to determine if aseptic transfer was used from the data provided, and three, be able to define the following terms: ubiquitous, aseptic transfer, inoculation, and a contaminant. In our local environment, microorganisms are ubiquitous, meaning they are essentially everywhere. This is a problem in the microbiology lab because we generally want to work with a pure culture, a culture that contains one and only one species. The reason for working with only one species is, or a pure culture, is when you have a tube and you have a result in the tube, like the tube media turned yellow, and you want to know, did your species do that or not? If you have a mixture of species in there and the tube turns yellow, you don't know if that's from the species of interest or from some other species in there. And if you only have one, two species in there and the tube turns yellow, then you know that the species is responsible for it turning yellow. Any questions about any of that? Louis Pasteur came up with the concept of pure cultures, and he developed a way of obtaining a pure culture, but it was a very difficult way of attaining a pure culture. He took liquid cultures, took a dilution, made several different dilutions, meaning there'd be 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000, etc. And then he would replica plate those dilutions so that he had 10 tubes of one dilution. If in one of the dilutions, only one of the 10 tubes had growth, Louis Pasteur correctly assumed that it was a pure culture because it started from likely one or a few cells. And that was correct, but it was a very difficult way to obtain a pure culture. because there had to be 9 tubes that were empty and then 1 tube that had growth growing in it. We'll talk a little bit later that Dr. Koch came up with an easier way of developing a pure culture. Louis Pasteur also came up with the concept of aseptic technique and developed aseptic technique to minimize the risk of culture contamination. We're going to learn about using aseptic technique in this lab. During inoculation, you should never put your test tube caps or Petri dish lid on the workbench. Are you guys seeing my screen?
Yes.
Thank you. Because earlier today, I went through probably about half the lab and somebody said, could we see your screen? And I said, you got to be kidding. I've been showing you my screen and you're telling me that you're not seeing it. And I went and looked at it and it said, your screen sharing is on pause. So I was sharing, it's just for some reason it paused. And I've never even known that you could have that as an option. Like when I come up here, I don't know if you guys can see that. I don't have the option for pausing it. So I don't know why the screen sharing was paused. But I wanted to make sure you guys can see this because I'm going to try and illustrate what we're talking about. You have a cap. You should never take the cap and put it down on the workbench because there's bacteria on the workbench. Why isn't that shrinking now? Oh, there it goes. And you don't get it inside the cap, but you get it on the edge of the cap. And then when you bring the edge of the cap over, if you can see this, to the tube, this is pretend to be my tube, the bacteria can fall off the edge of the cap, the lid, into the tube. And then the same thing about Petri dishes. Oh, I'll just leave it. I'll talk about it in the picture. If you take the cap off the Petri dish and then put the Petri dish down on the surface, bacteria can be on the edge of the Petri dish. So you should never put the cap or the edge of the Petri dish on the table, and then vice versa. You should never put it upside down on the table either, because things can fall out of the air and then go into the lid or the cap. And then when you put it over your container, the bacteria that are on the lid could then fall down into your dish or the tube. So you should always hold the lids and the caps. And this is showing you for a test tube cap, you should hold it with your pinky finger in one hand and the loop in the other fingers of that same hand. And the other hand should be holding the tube. And you should hold the tube at an angle. I don't know if you guys can see this. You should hold the tube at an angle so that when things are dropping down, there's less space to get it so that it can drop into the tube. So if you have the tube straight up, there's a very wide surface that something can fall in. If you hold it at an angle, there's less surface that it can fall in. Any question about that? Or holding the cap? The same thing with holding the Petri dish lid. I don't know if you guys can see this either, but you should not take it off the you should not take it off the dish. You should hold it so that it's partway open and then partway protecting the dish, as is shown here. Now, if something falls down from the air, it's most likely going to fall on the top of the lid, and who cares, because that's contaminated already. There's bacteria on the lid already. The only risk you have is if something falls directly in this region here, it would drop into the plate. But it's much less than if you took the whole plate open and exposed. Any question about any of that? So in one of the earlier questions, this is something that was brought out in an earlier lab, that you should hold the lid over the plate while you're streaking it, like there's streaking in there. And that way you're protecting most of the plate, not all of the plate, but most of the plate from airborne contamination. And then the second thing you should do is work quickly and efficiently so you have the lid open for as little time as possible. And then this lab tells you some other things you should do. Don't talk while you're transferring, or have a open cap. Sterilize the tools before use as well as after use. And then disinfect your workstation before and after you do your work. Now in our lab, you won't be using bare fingers because your hands will be having gloves, and that'll help somewhat too, as long as the gloves fit your hand well. You know, I've lost the ability to move that down. I don't, there it is. Huh, I can't get it to go down now. I'll go here. Oh, there it goes. In order to inoculate new sterile media, that means transfer cells into it, we must make sure that everything around us is sterilized and all of our tools are sterilized. So obviously only inoculate media that is sterilized, only use a stock culture. Excuse me, there's the stock culture, there's the media. That's sterile, meaning you don't have a unsterile stock culture. And then your tool, the wire loop, should be sterilized before use. How you sterilize it is you put it in the flame of a Bunsen burner or in the bacteria incinerator until the loop becomes red hot. And then you take the loop out, cool it, pick up your cells, and then transfer it to the sterile media. All right, know the terms of this lab? In this laboratory exercise, you're not going to be physically performing any of these activities because this will be an online lab, but you will be watching videos of these activities, and we expect you to know the material in these videos as well as the material in this lab, and you will be quizzed on them. We have a little video for you to watch on proper aseptic transfer techniques. So watch that. In procedure 1, you're going to be transferring sterile liquid, actually sterile water, using a glass serological pipette into 9 mils of media, sterile media. In procedure 2, you're going to be transferring a sterile paper disc using clean forceps and put that sterile paper disc in a tube of sterile media. Any question about any of that? So in procedure 1, you use aseptic technique to transfer 1 ml of sterile water using a glass pipette into a tube of nutrient broth. Now, when you're working with a glass pipette or a glass birette, for that matter, you'll note that the water does not flow across the surface evenly. It forms a meniscus like that, where it's higher on the edge and lower in the center. How you read the meniscus is from the bottom of the meniscus. You don't read the meniscus in the middle. or from the top? All right, let's go down to procedure 1. Here's the tube of media we're going to inoculate. You should look at the tube and determine whether that is cloudy or translucent or transparent. If it's cloudy, it has growth in it. And if it's transparent, it does not have growth in it. So clear is the word I was looking for. If the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was successful. If, on the other hand, the nutrient broth is cloudy, hazy, or turbid, then organisms are growing in the broth, and aseptic transfer was unsuccessful. You must use this table to answer some of the questions in this lab. And your answer has to be worded similar to the wording in the table. Like for procedure 1, you have to say whether the broth is clear or turbid, hazy, or cloudy. And the conclusion, you have to discuss whether the organisms are growing in the broth and or not, and whether aseptic transfer was successful or not. Any question about any of that? All right, here's the tube of procedure 1. After transferring the sterile water in the media and then incubating the tube overnight at the appropriate temperature, which is either 30 or 35 degrees Celsius, whatever the lab said. And there's a picture of it after incubation. Then answer for you, you'll have 0 there for question 0. Describe the initial clarity of the nutrient broth prior to inoculating the tube, both for the sterile water or for the paper disc. Discuss whether it's clear or turbid. So I'll tell you the answer is one of those two there. And there's the tube of broth. And then for procedure 1, you answer question 1. P1 stands for procedure 1. Describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees. State whether it's clear or turbid. And there's the tube there. Based on these results, what conclusion can be drawn? And you must use wording similar to the table. If you don't use wording similar to the table, I will mark it wrong. It doesn't have to be the exact wording. It just has to be similar. Then you must talk about whether there's growth in the tube or not, and if the aseptic transfer was successful or not. And then 1C lists 3 sources of error with aseptic transfer technique that could explain why the nutrient broth showed growth of microbes. 1, 2, 3. Any question about that? If not, let's go back to procedure 2. In procedure 2, you're using aseptic transfer technique to transfer a sterile paper disc into a nutrient broth. How you do that is we have a paper disc in a petri dish, which has been autoclaved, so the paper is sterile. And then you get some forceps, which are obviously not sterile, and you clean them. You don't want to sterilize them by sticking the forceps in the bacto incinerator, and then the tips of the forceps turn red hot, because the heat of the metal forceps will just go right up the forceps, and then you won't be able to hold them. Any question about that? So how you clean them is you put the tip of the forceps in the bacto incinerator just for a little bit. You don't get it red hot. You just heat them up a little bit, and then take them out, and then take the forceps and stick it in some alcohol. Take the forceps out and put it back in the Bacto incinerator. You should see the Bacto incinerator lighting the alcohol on the forceps. That will clean the forceps. It won't be sterile, but it will be good enough for this use, meaning there will be very little bacteria on the forceps. And then use these clean forceps to take a paper disc out of the Petri dish and then put the paper disc in the tube of media. Take the tube and obviously put the cap on and put it in the incubator for overnight incubation. Any questions about that? All right, there's the tube of media before you put the paper disc in it. And there's the paper disc, the tube after you put the paper disc in it. You can actually see it in the bottom of the tube. And this is after we've incubated it overnight at 35 degrees Celsius. Remember to use this table for procedure 2. If the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was successful. If the nutrient broth is turbid, hazy, or cloudy, organisms are growing in the broth, and aseptic transfer was unsuccessful. And then scroll down to question 2 and answer these questions. Describe the clarity of the nutrient broth after inoculating with sterile paper disc and overnight incubation at 35 degrees Celsius. Is it clear or turbid? Right there. And then based on this results, what conclusion can be drawn? Make sure you use wording similar to the table. For procedure 3, we're going to be transferring bacteria from a stock culture into a liquid broth. And we're going to be transferring Serratia marsans from the stock culture to a liquid broth, meaning a liquid media. Serratia marsans is a pigmented bacteria. It either grows pink in a broth, or if you grow it on solid media, it grows red. Here we have a video for both procedures 3 and 4, demonstrating how to correctly transfer from a stock culture to a sterile broth culture. And then for procedure 4, you're going to be transferring it to a sterile auger slant. And so that's shown in this video as well. Any question about any of that? All right, when you're watching this video, you only need to watch it into 4 minutes and 23 seconds of the video. After that point, you can stop the video if you wish. You should note that in the video, they're using the bacteria E. coli, which is an unpigmented bacterium. We are transferring Serratia marsans, which is pigmented. The procedures do not matter whether the bacteria is pigmented or not. And then you should catch an error in this video. The error is between 3 minutes and 5 seconds and 3 minutes and 20 seconds. Try and figure out what the error is. You can hear some sizzling when the instructor puts the loop into the E. coli. And that sizzling means something. And try and figure out what that error was. Okay? So Procedure 3, we're transferring Serratia mersens into a liquid broth culture. Serratia mersens in a liquid broth will appear pink. Broth cultures are problematic because it's difficult to determine if contamination has occurred. And this is because all the cells will mix together in the liquid broth. If you have minor contaminant, meaning where the contamination is the minority of the cells and the majority of the cells are the cells you're intending, you will not see the contaminant. The contaminant will be hidden by the cells that are supposed to be in there. However, if you have major contamination, where the contamination is more prevalent than the cells of interest, you will probably notice something wrong and figure out that the tube is contaminated. For example, if in this media you had Serratia mersens and E. coli, and E. coli was the majority of the cells growing in the tube, then the tube will not take on a pink appearance or will be very, very light pink because E. coli is unpigmented. However, if there's minor contamination, let's say E. coli is around 10% or anything really less than 50%, you won't notice it. The media will look pink because the majority of the cells are pink. Any question about that? All right. Use the table for procedure 3. If the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was unsuccessful. Can anyone explain to me why, if no organisms are growing in the broth, that aseptic transfer is unsuccessful here? Anyone gonna guess? Either you guys don't know or you guys are just very shy and nobody wants to risk being wrong. It's okay if you're wrong. That's how you learn, especially when I'm here to help you.
Is it because the bacterium is pigmented? We'll
Well,
be able to
yes,
see
but
it.
what are you doing with the bacterium? Where are you getting the bacteria and where are you putting the bacteria? You're not
I don't
fully
know.
understanding the lab here. Go on.
No, I'm not sure. I'll have to read through it again
All
to make
right.
sure I got
What you're doing is you're transferring from a stock culture and putting the bacteria into some sterile media. Okay. So that's what we're doing, meaning that we are purposely putting cells into the tube. And so if there's no organisms growing in the tube, then aseptic transfer was unsuccessful. And hopefully you understand that. If not, think about it. And if nutrient broth is turbid, hazy, or cloudy, organisms are growing in the broth and aseptic transfer was successful. For procedure 3, this is the tube. After transferring the cells into the media and inoculating the media or the tube overnight at, I think that one's at 30 degrees Celsius. Yep, 30 degrees Celsius. And then come down to the question for procedure 3, describe the clarity of the nutrient broth after inoculating with Serratia mersens and overnight incubation at 30 degrees. Was it clear or turbid? Take a look at the picture, answer it. Based on these results, what conclusion can be drawn? So come up to the table. for procedure 3 and answer what the conclusion is. And you can use the exact wording, but you can just use wording similar to the table, but it must be similar. All right. How do you know whether you've transferred the correct microbe, which is Serratia marsens? How do you know if you transferred the correct microbe? Anyone? All right, think about it. That's one of the questions. For procedure 4, we're transferring Serratia marsens to a sterile agar slant. Remember, Serratia marsens is pink in a liquid media or a broth, but when it's grown on a solid media, it's red in color. With an agar slant culture, or any solid culture for that matter, contamination can more easily be detected than in a broth. The reason being, here's a slant, and we, on the slant, take a loop and take our cells and then streak the bacteria in an S-like motion so that you get this type of a streak on the slant. We should see bacteria that is red on the slant. If ever you see bacteria off of the slant, like right here, and especially if it's not red in color, then you know that's a contaminant. Another way to tell if you have a contaminant is, let's say it's red all the way over here, but right there, this corner right there of the slant, it's not red. It's some other color, like white or off-white. Then this right here will be a contaminant. Ugh. And then come down to the tubes. P4 is procedure 4. And there you can see the bacteria. There's so much bacteria that it was streaked out like that, but you can't see the streak. It's just growing all the way there. Right there, there's a little bit there, but mostly right there, you can see some agar that was not streaked, and there's no bacteria growing on it. Scroll down to the questions. Question number 4 for procedure 4. Is there bacterial growth on the slant? All you need to say is yes or no. And then for B, how do you know that there's bacterial growth on the slant? And you just give your answer, okay? And then for C, does it appear to be the bacterium you inoculated or a contaminant? And I suppose you should say a little bit why. And there it is there. The bacteria has the correct color, so it looks like it's the correct species. And it looks everywhere the same. I guess I should mention a little bit there. Right here, it's a little bit lighter, but that's because there's some water in the bottom of the tube in the slant. All right, that's it for this lab. Are there any questions? If not, let me state that I have graded lab 0 and lab 1. And if you didn't get 100% by this Saturday, you can submit a correction for 100%. And some of you didn't do too well. You need to think a little bit more about the lab or get a little more help in the lab from me or someone else.
I have a question. I was wondering if on the comments for my first lab that I turned in, you said you allowed one air so that it was okay. Do you allow one air just for the first lab or for all of the labs?
Uh, it's for the first grading of all the labs.
Okay.
So if you make one
Okay.
error in the first grading, I will allow it and you'll get full credit. However, if you make more than one error, then you will obviously have the penalty and you can correct that with the second grading.
Okay, thank you.
Okay. All right, I will be here to answer any questions until about 8 o'clock, but I will log off when the last student logs off. But I am here to help you if you have any questions. So use me. That's why I'm here for.